conference. And I'm going to talk about the first chapter of my PhD. I'm a PhD student at the Mediterranean Institute of Advanced Studies in Mallorca. And what we did is we gathered manual data with focal observations and also with cameras of the same study sites in a natural environment, and we compare the data set. Then I don't want to convince anybody here that there is a worldwide pollinator crisis. And what we do in our lab is we study at the ecological point of view. When we study site, we don't focus in any specific group of plant or pollinator. We study the whole network, how they interact among them, and which metrics we can extract from those interactions. And then we usually work on islands. Then we can compare islands that are invaded by invasive species or not. Then this data that we gather, we obtain it with focal observations. We gather among 1 hour or two per plant species, depending on the abundance. The species that we don't know how to identify it in the field, we collect it and we bring it to the entomologist. And yeah, but we all know that these methodologies have limitations. For us, one I want to highlight is that sometimes we also work on islets. So they are difficult to reach those places. So when we are there, we want to extend the temporal and the spatial resolution of our data set. So what we did, is to put a lot of cameras in wild plants. So also another benefit from this methodology is that when we have interactions, we have the manual notes in our fieldwork, but now with the cameras, we can recheck which species has visited which flower if that interaction happened there. And also is an opportunity, as we saw before in the talk before, that you can study new insights. So the first chapter is only about what kind of data can provide the cameras, and if it's complementary or which are the differences or the biases of both data sets. So what we hypothesize is that as the cameras stay longer periods in front of the same individual, we'll take more low frequent interactions, that we can see differences. In the islands, we have lizard pollinators. I say in the islands because it's an island phenomenon around the world. So it doesn't happen in the mainland, but yes, in the island. So we work on islands, so we suppose the human presence will disturb this lizard pollination. And we suppose that we will have similar core of interactions and similar network metrics. To shorten the process of the video reviewing of the cameras, we did an algorithm to only review the presence of the video. So we only did detection or classification, and then the species classification was made manually. I started doing some field tests, putting the cameras in Tupperwares, but it was not enough to have good quality data. So we go for focal lengths with a 3D printed case. We pulled all the components. We put it in the external case for the environment, for protected for the environment. And with the tripod, we have the facility to point to a tree or to a plant in the floor or in different methods. So this is how it looks. And we did it during the night and the day. And we got those detections during the day and the night. However, this chapter is only comparing the diurnal data with both data sets. So the algorithm performed quite well with the high-Q quality camera of the Raspberry Pi. So we relied on those detections. This is how it looks. This is the quality of the video. And also, you can see that it detects as a pollinator different morphotypes. Microdiptera here. This is an Anthophora hispanica and the Coletes B, medium-sized B. Also, it has some false positives, but they were not a problem because, well, we prefer false positive because we don't want to lose interactions and information in this case. To do this, we selected a tiny island because it is a simple ecosystem. So we had enough cameras to do this, and we did it in 6 study sites. We did 13 visits between 2 years, 2 spring seasons. Each visit contains 6 days of fieldwork, and in always we were 2 field technicians and 2 camera per plant species, so 2 individuals per plant species with a maximum of 6 flowering plants. We had 12 cameras. I'm not going to the details of the formulas that we do with the networks, but the statistical comparison was done with the 13 visits, the 6 study sites, and the 2 methods. And the results and the discussion, well, we had no different significance with the species richness and the abundance and the Shannon diversity. We had similar results with the previous talk that we per pollinator species, we had more detections when we standardized the data set with automatic monitoring in front of direct observations. When we compare this per plant species, we had some drops in the automatic camera system that could be explaining that when the human is observing the flowers, the lizard is visiting less the flower, then the insects visit more the flower. So we have some drops compared to the human observations. However, when we plot this per unit of flower, we had similar performance of both methods. Here we compare the metrics of the networks. Maybe you are not familiarized with these metrics. The connectance calculates the overall rate of the connections within all the possible connections, how many connections have appeared in the data set. So the connectance was higher with the automatic camera system because it provides more rare frequent interactions, more low frequent interactions, staying more time in the same plant. This makes more connections among all the probabilities and more network specialization, that it means those specialized interactions that one specific bee goes only to one kind of flower. So they were more detected with the automatic camera system. That's why it's a bit increased. And the robustness is lower because those specific interactions are more prone to disappear. So we plotted the networks. We have sorted in the equal way the pollinators here, and here the plant species are sorted equally. And we can see that the general picture is similar for both data sets. However, we plotted the red dots. So as more dark, more frequent is the interaction, right? So the picture as it's sorted equally is similar. But we plotted the missing interactions of each method, taking the other one as a ground truth. So we plotted those interactions. So we did this plot. We plot all the pace of interactions here, and don't look at the red dots now, only in the gray. So we plotted here the detection with the direct observation and without automatic monitoring. And then here in yellow, we have those interactions that they were not detected with the direct observation. So we wanted to count how many pairs of interactions, unique interactions, has missed the direct observations. Then we plot it in the other axis, and we saw when it was only detected close to 1 time with automatic monitoring, there were 30 interactions missed by the direct observations. But as more frequent as those interactions are less, there are less interactions missed by the direct observations. In resume, this is just to prove that those low frequent interactions were more detected by the automatic monitoring. It's a bit confusing, that graph. And when we plot the interactions that the automatic camera system missed, we saw that it was about the insect size. Here we have an exception because this pollinator Sysphida went a lot to a plant with human observations. So it was bigger than 5 millimeters, but most of them under 5 millimeters were less detected by the automatic camera systems. That's why in the first graph I show you there were some detections only detected by the direct observations. Well, result summary is we had similar core of interaction, similar species richness, similar abundance, some differences. We hypothesized that we were not going to have those differences, but we have it in the network metrics. And the focal observations still outperform the detecting small body insects, highlighting an area for future refinement. And as a take-home message, this algorithm should be improved. And there is, it can be a lot of work that it can be done. The human presence influence the pollinator behavior, and focal observations can potentially underestimate those low-frequency interactions. And they are ideal for long-term and remote deployments. And in my PhD, this is my first chapter, so I have to do 2 more at least to defend. And now I'm going to start a collaboration with Aarhus University to go more into general classification. I don't know if you saw the Teams talk yesterday. And at some point I would like to automatize the interaction. So to detect those interactions automatically, not only the pollinator, and also to have the tracking. And at the end, what I want to extract from these methodologies is ecological information. And and in my next studies, I'm going to compare invaded and uninvaded islands by Apis mellifera. So let's sit.
Thank you, Paul. Questions?
Luca?
Yeah. Can you go back a second to the size comparison?
This one?
No.
Wait.
3 slides back.
Yeah. Yeah. You don't have the automatic detections here with the insect size. This is only.
No, this is insect site here, and the detection by the observations, and the missing counts are represented like this.
Yeah.
With the circle and the hit.
My question, however, was about something different.
Okay.
Presumably, you have very different sampling sizes for the networks between the automatic counts and the manual observations. Like the sample size, like the number of observations you have is very different.
It's smaller by the camera.
Yeah. So did you subsample these networks to have an equal sample size, because the network metrics depend on how many observations you do, or how did you give?
It's all about how many flowers you can control, right?
Yeah.
What we did is standardize the data per time and flower unit.
Okay.
So we standardize on the data.
So I have the same time investment.
Exactly.
I've got 2 questions. One is to do with hardware, one is to do with algorithms. First one, how much did your self-made device cost, and did it require SD card storage, or were you able to transfer the data directly? And the second one is, I think you said you used LSTM. Was it LSTM on its own, or was it a hybrid CNN LSTM system?
The LSTM was to improve the detection of the.
Because normally LSTMs are used for time sequence stuff rather than image frames.
Yeah.
So was that to do with tracking or something like that?
We did not tracking. It was just to improve the detection in all the frames of the video.
But it wasn't just LSTM, was it?
No.
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 I was going to say, I'm surprised it was just LSTM.
No.
And the cost per unit, and whether or not you needed to.
Here the components. This is a commercial power bank that you can buy for your phone. So it's around 20 euros, let's say. The Raspberry Pi is increasing now the price because of the war and the stock and whatever, but at that time was 60 euros more or less. This camera is 70 euros. When you print this, it also depends on where you go to print it, because I know that now there is a group in Madrid that they are replicating the same system and they pay less than us because they found a better place. But we paid 60 euros for this and almost 100 for the external case with the cover.
Close to 300 euros per unit.
Yeah, close to it. But I think
That's cheaper than you could get a commercial one and do the same job.
the thing is, here is not that slide. What I wanted to, yesterday I showed it in the Harvard demo. What we need is to schedule the recordings, because we need to monitor a full morning, a full afternoon. Then we put it in the night. So we need these periods of recording, stop, recording, stop, recording, stop. So with the commercial camera, you can start recording, but then, yeah. And also you have other possibilities with the Raspberry Pi, no? You can, instead of recording 24 frames per second, you can record 10 frames per second and maybe have the same information. I don't know. I did it with 24, but.
But commercial systems don't achieve this battery life.
Also.
That's
And if it's a short one, please, we have.
so thanks so much for sharing.
No, if you look in the video, as we sample those communities, we also capture insects. We identify all the diversity of those sites, and when we have the frames, we try to classify the frames until species level. As we were working in this tiny island, was quite achievable. Doing this in mainland maybe it's more difficult.
When you compare network methods, they are kind of standardized.
Yeah, they are. Yeah, the comparison is with the same pollinator species.
Thank you, Pam.